I'm going to talk about antigen antibody interaction. First, before I discuss about the antigen antibody interaction, I will briefly tell you about the antigen and the antibody. Then I will talk about the mechanism of interaction. So, antigens are broadly classified into two types, that is, particulate antigens and soluble antigens. The particulate antigens are bacterial cell, viral particle, cells. Cells could be bone marrow cells or cells of any tissue origin. Basically, the particulate antigen has a shape because a cell has an outer boundary and cytoplasm or outer boundary with some nucleic acid inside, like as in the case of viral particle. So, since cells are made up of different organic molecules, the surface of the cell or the molecules that are within the cell become the antigens. So, therefore, those antigens are called as particulate antigens. The soluble antigens are those which could be simple proteins, glycoproteins, glycolipids, or nucleoproteins. These could be the components from the cellular origin, and they could be present in the aqueous medium as soluble components. Altogether, the antigens are organic molecules. Similarly, the antibodies are produced in the body against the antigens by specialized cells as a defense mechanism, and these antibodies are always glycoproteins and found in the serum or the plasma of a person, and they'll be circulating in the body, and whenever they come in contact with the antigen, they bind to the antigen, similar to the mechanism how the enzyme binds to the substrate. Overall, if you say the antigen antibody interaction, it is just like a protein-protein interaction or like an enzyme substrate interaction. Generally, since the antibodies are soluble organic molecules, they can react with particulate antigens to form an agglutinated mass, or they can react with soluble antigens to form a precipitate, and that reaction is called as precipitin reaction. And when they form the agglutination of the particulate antigen, it is called as agglutination reaction. Just to give an example of antigens, on the bacterial cell, you can see that the antigen could be consisting of the mucopolysaccharide capsid surrounding the plasma membrane of the bacteria, which is called as VI antigen in this diagram. Similarly, it could be membrane bound antigen, which could be called as O antigen, and the flagella of the bacteria, which is called as flagellin, is called as H antigen. All these are present in the bacteria, could be recognized as antigenic epitopes or determiners, and antibodies will be produced against these individually and recognize these molecules like H antigen or O antigen or VI antigen as independent entities by the antibody and cause agglutination of the bacterial cell. Then, if we take a typical viral particle, the viral particle will have the spike protein, then it could have the membrane and the membrane proteins as M, and then the nucleoproteins in the form of nucleic acid associated with the protein, then the enzyme that is associated with the membrane for the virus to enter the host cell, like hemagglutinin esterase here in this example, and the envelope small membrane proteins which are concerned with the envelope or the protein coat of the virus, like this. Now, each of these, once again, in the virus could help as antigens and recognized as antigens and antibody produced against the spike protein will not be reacting with membrane protein. So membrane protein antigen will be different in chemical characteristics compared to the spike protein. So that means spike protein, glycoprotein, acts as one antigenic determiner, membrane protein acts as another antigenic determiner. So nucleoproteins will act as another antigenic determiner, like that. So antibodies produced against the virus will be against these antigens at any given time when the virus infects a person. Therefore, many antibodies of different types against a particular antigen. Antigen as a whole, now you say virus itself is an antigen, which is a particulate antigen consisting of different chemical components and each of them acting as antigens, stimulating the antibody production. So therefore, those antibodies will be in different types. So typically, then antigen antibody reaction takes place due to chemical interaction. Now here on your left hand side, you have antigen molecule. On the right hand side, antibody molecule. So the interactions that typically take place between antigen antibody have been shown by chemical interaction. The first simplest type of interaction that could take place is by hydrogen bonding, because both these molecules are organic. They would definitely contain OH groups, COH groups, okay, H as an exposed element. So because of that, the OH reaction or OH-H reaction would result in hydrogen bonding. Then there could be possibility of NH2 groups or NH3 groups exposed which interact with the COO group. That will result in the formation of a salt bridge. COO group reacting with NH3 group, forming the salt bridge, which is called as ionic bond. Ionic bond is the strongest bond that you can see between the antigen and the antibody. One thing you note, there is no covalent interaction in the antigen antibody interaction. Whatever the interactions that take place are weak interactions, but ionic bond is the strongest bond that could take place between antigen and the antibody. Similarly, most weakest interaction would be hydrophobic interaction, where hydrophobic groups which are exposed on the surface of the antibody and the antigen could interact with one another, forming a hydrosphere around them, and that would be hydrophobic interaction. Here they have shown interaction between CH3 groups of both antigen and the antibody. Similarly, it could involve simple van der Waals forces or Coulomb's forces, which are also weaker compared to ionic bond. So, mechanism of interaction between antigen and antibody is nothing but chemical interaction between the functional chemical groups that are present in the antigen and the antibody. As I had mentioned earlier, the antigen could be a protein, or it could be a glycoprotein or nucleoprotein. Okay, so they would definitely contain the functional groups like OH, NH3, CH3, COH, all these groups. Similarly, antibody being a glycoprotein, it would have amino acids, and those amino acids could have COO group exposed or NH2 group exposed, and these are the interaction that facilitates the antigen antibody interaction, which is a chemical interaction.